let's see what is the relevance to industry of plant cell technology. So talking about some success stories, see this is one of the plants, Silene stenophila plant, which was found, the seeds were found in the permafrost conditions in Siberian region. So in the fossils of one squirrel burrow. So these seeds could be then lead to, through in vitro plant cell cultivation techniques, could be revived, and that is the plant which you can see. So that's the application of plant cell and tissue culture, that such, and the age old was 30,000 years old permafrost conditions. So in this region the seeds could be obtained, and then the seeds could be revived to lead to a whole plant. So now it is a live plant which is giving rise to new progenies. Similarly, it is plant cell and tissue culture can also be used for conservation of endangered plants and rare plant species. So some of the plants which are very rare and are under endangered list, those can be revived and can be conserved using plant cell and tissue culture techniques. So this is Ilex khasiana, that is a critically endangered plant in northeastern region of India. So that was able to be conserved using synthetic seeds and plant tissue culture technology. In order to cater to the increasing demand in the market, whether it is for agricultural crops, whether it is for medicinal compounds, you need artificial seeds. So plant cell and tissue culture can also be used for production of artificial seeds if you want to do large scale shoot multiplication. So this is how it is done. Cell immobilization techniques, where the embryo which is generated using somatic cells, we will be talking about it in detail, is encapsulated in gel beads, and these can be used as seeds to lead to plants. So rapidly multiplying stalks of the plants, so large number of plants can be regenerated through these techniques. Now you all know about GM crops, genetically modified crops. In order to improve either the biomass productivity, the growth, or the production of that medicinal compounds, genetic modification is done. And so one of the most successful example is the BT cotton. So almost 60% increase was found in the productivities is by bringing in the BT cotton. So have you heard about bioplastics? So up till more than 50% dry cell weight basis, people have been able to express PHBs, polyhydroxyalkanoids, in plants. So there is an opportunity to produce bioplastics in plants itself. So where the plants like switchgrass, which are rapidly multiplying plants or weeds, can be used to produce as stocks for feedstocks for bioplastics. So that's again through plant cell technology. Anti-cancer drugs, one of the most popular drugs are vincristine, vinblastine and Taxol. So that is this Taxol, it is an anti-cancer drug which is produced from yew tree. So and we are in the lab are also working with camptothecin. Camptothecin is another lead molecule. In India we use it for irinotecan, topotecan for lung cancer, cervical cancer. It's marketed drug. So that is obtained from in India from Nothapodites pneumoniana. That's again an endangered plant now in Western Ghats because of rapid uprooting by the industries. So plants again are valuable stocks for high value medicinal compounds. So now, as I said, when you use natural plants for extraction of such high value molecules, what are the disadvantages? What can be the disadvantages?
Yeah, extension of that species.
You may lead to endangered plant, then plant getting endangered. What other disadvantage? It may not be enough to sufficient to cater to the demand. Okay, what else? What else do you think can be a demerit in natural plant extraction? Hard in the sense, if I would put it in a sense that the downstream processing is more difficult, why? Very true. Because you are extracting it from the whole plant, the plant will have n number of molecules. Okay, what else? If there is seasonal variation, and along with seasonal there can be geographic variation. See, we ourselves, when we were working with one of the projects which is on cyclic peptides called cyclotides in the lab. So these are small mini proteins from plants which have plethora of therapeutic activities and potential. So when we were looking at it, we collected plants one from the Himalayan region, one from the Ooty region, and some of the plants we brought and we cultivated them in the horticulture at IIT Madras. For 3 years we cultivated them, and after 3 years we began the study of characterizing the different types of cyclotides, these mini proteins, and the kind of cyclotides which are present in the plant material. Interestingly, what we found was that the plant's material which was brought from Himalaya, the same variety, the one which was brought from Ooty and the one which was brought from Ooty but cultivated for 3 years, had very different array of cyclotides. Now the plants were the same which were being cultivated in Ooty, and we brought them to IIT Madras. So that shows that there is high impact of climatic and geographic variability. And these are a class of secondary metabolites. So they are defense related compounds. So even the microflora which is present around in the soil impacts the kind of secondary metabolite or the expression of secondary metabolites in the plant. So that showed us as there was a difference in the aerial parts to ground parts. There was a difference in different parts of the plant. There was a difference in the kind of certain novel cyclotides came out which were not present in the other varieties in Chennai. And the plants which were those there in the Ooty could not show those novel cyclotides which we could detect when we cultivated them in Chennai for 3 years. So that shows that now an industry cannot work at this pattern. It depends on product quality. Product quality means there has to be consistent product quality and quantity. So now for consistent product quality and quantity, natural plant extraction has a demerit associated because there can be variation, as she said, with season. Some of the plants, for example, azadirachtin, it's a biopesticide. It is obtained from neem. Now and majorly from the part which has its maximum content, which is the seed. So now all throughout the year, seeds are not available. They are only available twice in a year. So now for twice in a year or once in a year, one will have to wait for that year long and then collect the seeds. So there is demand and supply gap which can crop up. So an in vitro cultivation technique will have therefore higher productivity because with respect to time and moreover less space and time requirement. So that's what we were talking about. Then the available land, because of the population increase, there has to be a priority drawn between agricultural crops, medicinal plants. So there is a gap in the available land. Then low concentration of the product, because as you people were saying, downstream processing is difficult because there are n number of molecules present in the plant. Now imagine the same carbon flux is being diverted towards array of different metabolites. Under in vitro conditions, you have the plant cells are amenable to process optimization. Now process optimization, which means that you can divert desirably the metabolic flux towards your desired compound. Then limited and non-uniform supply. This is because of the climatic and geographical variability in nature. So therefore, what can be an alternative strategy? An alternative strategy is like using plant cell technology, where under controlled environment, if you grow the plant cells and you force them to produce your desired metabolite. That is the advantage of plant cell and tissue culture. So what is plant cell technology? Plant cell technology, there are different forms of in vitro cultures which come under plant cell technology. You can do cell culture, you can do organ culture. Organ culture can include your somatic embryos or your roots, simple roots, multiplying the roots only, or multiplying shoots, just continue to multiply shoot, or hairy roots, agrobacterium transformed cultures. So these can be different forms of cultures which can be used for large scale production of the high value chemicals. So as a definition, what does it mean? The culture of plant seeds, organ, tissues, cells or protoplasts. Now what are protoplasts? Devoid of the cell wall. So sometimes protoplasts are required. It becomes easier to manipulate and do hybridization with protoplasts because cell wall has been removed. So what is so different about plant cells which has been exploited in plant cell technology and peculiar about plant cells? A very good advantage, which is totipotency. Now what is totipotency? Totipotency comes from the theory a cell from cell. So cell theory everybody knows. So all the genetic information is present in the cell. So totipotency is the ability of the plant cell, any plant cell, to reverse, even if it is organized and performing a specialized function, it can reverse back to its multiplication stage and again reprogram its genetic information to reorganize itself into some other specialized function. That is what is totipotency. So the potential or the inherent capacity of the plant cell to develop into an entire plant. So even from single cell, you can regenerate by rapid multiplication and reprogramming of the genetic machinery into an entire plant. Then it implies that all the information which is required to generate into a plant is present in the single cell. It is just that not all information is being used at a given time point. The cell has all its information, but some cells are performing one function, some cells are performing the other function. It doesn't mean that it doesn't have the genetic machinery, it's about expression. Whether the cell is expressing all the genetic information or not. This is what we also found with coming back to the giving you an example on cyclotides. Why were we able to find novel cyclotides in the same plant variety? Although it was the same plant which was cultivated from Ooty, transferred to IIT Madras. Suddenly from where is the novel cyclotide coming? Which means that the genetic machinery must have been there but must have been encrypted. It is the epigenetic factors or the environmental conditions which might have been able to induce the expression of these silent genes. So now, how some idea about the history. It began in parallel around the World War times, both in Europe, in US. They could continuously culture the carrot cells in the medium and keep it live. That was the first breakthrough. And then later they also found they were using coconut milk, and they also found that there is a compound in the coconut milk. The first auxin to be discovered was IAA, indole acetic acid, which was said to be playing a huge role in expression and keeping the cells live. But everybody then started thinking that IAA is the compound used for in vitro cultures. But when coconut milk also was being used, with this advent of your microscopy and your analytical instruments, and it was also found that apart from auxin, there is some other compound which is not IAA, auxin, present in the coconut milk, which is significantly affecting the in vitro cultures. So that then came the discovery of kinetins. So that's how auxins and kinetins came into the picture. So the initial discovery and all the inventions which took place in plant cell technology dealt with the nutrient requirements of plant cells, then the different forms of cultures, what can lead to roots, what can lead to shoots, which means auxins and cytokinins. So there was a lot of discovery of hormones and importance of hormones which took place. Then later people came out with nutrient requirements. People also came out with predefined medium compositions, MS media, then Gambogg's mediums. So these are varying in nutrients, sometimes in hormones presence, nitrogen source in some of the media it is present, in some media the nitrogen is not present. So depending on what kind of culture, in vitro cultures, you would like to induce, some of these predefined media work well irrespective of the species on which you are using. So MS, Gambors, these are very popularly, or White's medium, these are very popularly used predefined nutrient media for in vitro cultures depending on the objective. Whether you want to do root culture, shoot culture or callus, these are used. I was talking about the hormones. So basically there are other hormones also, gibberellins, then abscisic acid. So, but two major hormones which impact the in vitro cultivations are auxins and cytokinins. Auxins are said to be root promoting hormones and cytokinins are shoot promoting hormones. So generally this is the general trend, but sometimes may vary. So general trend is that if you vary and keep the auxins and cytokinins concentration high, it may lead to dedifferentiation of the explant, which may lead to callus formation. So what all things which will impact the plant cell and tissue culture? The explant source. What kind of explant use? Not all explants may lead to the in vitro culture which you would like to. So type of explant which you use also matters. So which can include any plant parts, especially young plants parts are generally used for in vitro cultures. Why do you think? They are more amenable to changes, reprogramming. Medium composition, carbon and nitrogen source. Then aseptic conditions would be essential. Environmental conditions, plant cells, so light plays a major role, and rest as any other fermentation. Now genetics in the sense that if you think that using this particular auxin and that and a particular kinin, cytokinin is leading to callus generation or root generation in sunflower may not work with Viola odorata. So it is species dependent, what kind of hormones, therefore every time that optimization of what kind of hormones and their concentration is important. So this is a small flow chart of how plant cell fermentation is done. You start with the plant either in vitro, inside developed shoots which I showed you like in vitro Viola odorata, or outside. But outside explants you need to do surface sterilization, which means from the surface the contaminants will have to be removed. And then you bring it to a dedifferentiated form which is called as callus. After you have callus, you put it in suspension so that you disperse the cells and it becomes a fine suspension. You need to subculture and filter so that the aggregates are removed. Now, as why did I say that you filter and then subculture? There can be number of subculture cycles required in inoculum preparation or in suspension culture. This will facilitate what? Filtering and bringing the cells uniform. This uniformity is with respect to in terms of cell synchronization. Very nice. So this leads to synchronous culture. If you pass through cell sieves of uniform some size continuously, you can assume that whatever passes through will be uniform in size and also therefore may be more uniform and more synchronous even in their metabolic activity. So gradually you may lead to synchronous cultures, shape and size. And then you bring it to cell suspension cultures. Once you have cell suspension cultures, then we implement all optimization strategies. What kind of optimization strategies? I'll just brief you. And then you bring it to the reactor level where all scale up strategies are designed before you do the downstream. So as I was talking about, how do you begin? You take the plant parts and then on the solidified medium with predefined hormonal conditions, it will dedifferentiate into a callus form. This callus is transferred into a liquid medium of desired composition with hormones and you get the suspension culture. So this is also one of the endangered plants for whom the callus could be generated. This is from a northeastern region. So let's talk about brief you on the different strategies which we will be learning to optimize the plant cell bioprocess. Once which is same as bacterial fermentations or microbial fermentations, we will be optimizing the media. What techniques generally we use to optimize the media? If you are given to optimize the medium composition, how would you optimize it? How would you optimize? The objective is?
Use one factor at a time approach.
What else?
We can change the concentration of each of the components at a time. Like carbon source, we can supply like 1%, 2% accordingly to understand how much of the carbon source is required by the plant.
Okay.
We can do for the nitrogen source and for the
Okay.
salts and all.
So there will be some 16 to 18 nutrients. So each nutrient will vary in a range, 1%, 2%, up to what? Up to some 10, 20, 30. So how many number of experiments then? 18 nutrients with, so how many permutations and combinations? Hard to even imagine. So my whole PhD will go only optimizing the media, isn't it? And plant cells grow slow. One single experiment will take around 15 days minimum. 15 days. So imagine the number of experiments and the amount of time you will be spending in optimizing the media. Any other better idea? This is doable. I'm not saying this is wrong. Okay.
Statistical techniques also.
Response surface management.
How will that help?
For optimization, we need to check different compositions, and those statistical techniques may give us different compositions, and we can run those.
How? Some more detail. Statistical technique only doesn't help. How? Raji, she's right. She's right that statistical techniques can be used. But how are they useful? In what way will they help single factor?
We can identify the most
Wonderful,
critical ones.
very nice. So first thing is, out of these 16, a smart person would be to first identify where should I spend my time and money. I need not even optimize something which is not impacting the results so much. So first I will try to identify out of the n number of parameters which I think are present are critically or most significantly impacting. So I need the ranking to be done. So statistical optimization, it helps in reducing the number of experiments, and in those minimum number of experiments, it can help you to identify the most significant ones, and then after I have identified the most significant ones, then what should I do? Again, the statistical optimization, design of experiments can be used. It will help in what? You were talking about response surface methodology. So what is response surface methodology?
So we will do the high value and high level, low level of that composition. We will try the combination combined effects.
Okay. So response surface methodology is one of the ways. These are nothing but factorial designs. Now what are factorial experiments? Now full factorial, or they are partial. Some of the there is an entire design space. Once for every parameter, there will be, from where the single factor experiments will be useful is if you want to know the range. You must widen your range as far as possible so that you know the minus and the plus level, where you can be sure that there will be an inflection point. There's no point in selecting a range where there is no inflection point. How will it maximize the objective function? Isn't it? So your single factor experiments are useful to identify the inflection, though a range where you are definite to find an inflection point. Now a range where you are definite to find an inflection point is what? You must have a small value and a high value such that one works positively and the other is less. So some 3 points such that, so that you can identify that yes, there was a hill. Some single factor experiments should be selected in that manner. Once you have identified the positive and the negative end, then you use that in your statistical design of experiments. Now there can be n number of permutation combinations even in this plus minus range. For all 18, suppose you have selected 4 most significantly affecting. Even in those 4s, selecting minus a and plus a, you will have n number of permutations and combinations. Now, even in that, these design of experiments, they, based on statistics, they will select only few data points, recipe, and they will give you a recipe of your experiments. That suppose A, B, C, D were 4 factors. So this is how these 4 factors you carry out the experiment. Then they help you to identify how A, B, C, D is interacting with each other and how is it impacting the your objective function, which may be biomass or your product yield. Once you have identified that interactive effect taken into account, it will help you to optimize and predict the most optimum ABCD value in the range which you have given, which will lead to maximum growth or maximum product formation, depending on what objective function you have used. So that's how media optimization is generally done, which in less amount of time with minimum number of experiments can give you better results. Now, optimization of environmental factors also, because single factor optimizations, you all must be knowing, they don't take into account the interactive effect. But your statistical optimizations can take into account the interactive effect, therefore can give you the real optimized value. Addition of precursors. Now, this is another well-known strategy. Precursors are intermediates in the biosynthetic pathway. For example, let's take camptothecin or any Taxol. So, there are biosynthetic pathways involved where there will be n number of intermediates connecting to the central metabolism, that's secondary metabolism. Now, there will be n number of intermediates in that pathway. Which intermediate to add? Or adding any intermediates there which is close to your desired product can lead to increase in the rate of forward reaction to the desired product. So that is what is done. You select an intermediate which can be after exogenous, not all intermediates if added exogenously will be readily taken up by the cell, may become toxic also. So those intermediates which can be made available to the cell to be taken up inside the cell and get utilized will lead to enhancement in the rate of forward reaction and therefore leading to increased product formation. That is what is precursor addition. Now, permeability enhancers, one was precursor addition. Then you can even enhance your yield, product yield, by adding permeability enhancers. So how do you think permeability enhancers will increase the product yield? Permeability enhancers are what? They are compounds which can reversibly permeabilize the cell. Reversibly permeabilizing means that the cell viability is not getting impacted such that they become non-viable.
They increase mass transfer.
So because you are disturbing the permeability, so there would be more to and fro motion across the cell membrane, which can increase the mass transfer across the cell membrane. What else? So increasing the mass transfer, how will it improve the product yield?
The biomass will increase, so the product will be more.
The required component, the limiting
Immediate
components will be more available now.
For product formation. One is the nutrients can come and cause, but what else which can have an immediate impact? What can drive the rate of forward reaction? If you can remove the product, can make a sink for the product. So by permeabilizing the cell, the product is being let out, so which can form a sink for the product and thereby driving the reaction forward. So that's how permeability enhancers give an impact. Now elicitors, this is the most promising strategy which is used to improve the product yield in plant cell technology. Elicitors, these are molecules which stimulate defense or stress related responses in plants. Now majority of these high value compounds, as I said, are phytoalexins, are defense related compounds. So if you can exogenously add such molecules which can elicit or induce defense responses in the plant, be it as a signaling molecule which can lead to a cascade effect, thereby leading to an enhancement in the secondary metabolism, or something for which the plant is already immune to increase its defense response, maybe fungal components or any pathogenic component which can give the signal to the plant cell and induce the defense machinery. Plant cell immobilization is also used to improve the productivity. So these are some advantages. So one was, we were talking about, if suppose the species is shear sensitive, it forms aggregates, then it is useful. You have immobilized the cells, so the encapsulation helps it to overcome the shear forces.